mitochondrial genome evolution. How the genome of the mitochondria evolved. If we see the difference between the genome of mitochondria which is present in different species, we see that there is a large variation in the size of the mitochondrial genomes. Usually we see that the genome is less in size in case of animals and it is large in size in case of plants. There is no recombination events in the genome of the mitochondria. There are a little intergenic DNA. There are almost no introns, particularly in the case of animals, but in case of plants, some introns are found. A variable control region on its own genetic code. So these are some of the characteristics of mitochondrial genome. Reclinomonas americana is considered as the ancestral mitochondrial genome. This mitochondrial genome of Reclinomonas americana has total of 97 genes. All the protein coding genes are found in the mitochondrial DNA. There are 18 protein genes unique to this species. There are 4 genes encode a eubacteria-like RNA polymerase. There is vestigial prokaryotic operon organization, and there is universal genetic code which is used by this species. If we do a comparison between the ancestral and the derived mitochondrial genome, the ancestral mitochondrial genome had many genes, bacteria-like rRNA genes, ribosomal RNA genes. They have complete set of transfer RNAs, mostly coding sequences, gene clusters, and standard genetic code. So these were the characteristics of the ancestral mitochondrial genome. What are the characteristics of the derived? Loss of genes in many mitochondria, divergent ribosomal RNA and DNA, high mutation rate, non-coding DNA, biased codon usage, and non-standard genetic code. So these are the difference between the ancestral and the derived mitochondrial genomes. Mitochondrial phylogeny are separate origin for plant mitochondrial genome. In case of plant mitochondrial genome, the presence of unique 5 sRNA gene in plant mitochondria and structural difference led to the theory of separate origin of mitochondria in case of plants. Analysis of mitochondrial proteins gave topology similar to nuclear gene derived phylogenies. This is the phylogeny of the mitochondrial genome. Here we can see that these species, they have almost similar type of the mitochondrial genome. So their mitochondrial genome have a similarity with each other. In case of these species, their mitochondrial genome have a similarity with each other. So these are those species where the mitochondrial genome of these species, they have similarity with each other. Origin of mitochondria. Here we can see that this is phylogenetic analysis. In this phylogenetic analysis, species in yellow lack mitochondria. So here Entamoeba that lack mitochondria, and these are the species which also lack mitochondria. This phylogenetic analysis suggests that mitochondria were gained at position B. So at this position, those species, they attained they gained the mitochondria. So if we conclude that about the evolution of the genome of mitochondria, mitochondria seems to be monophyletic in origin, derived from a common proto-mitochondrial ancestor.